Hello everyone, welcome to today's biology practical session. In today's session, we will learn about the vertical section of mammalian blastula. Vertical section is a section that is taken on a vertical plane. The diagram that you're looking at here is actually a vertical section. What that means is this structure is actually like a sphere, and what you're looking at here is a section of that. When you cut it vertically like this is what it would appear like. Why we are doing this is to observe what are the structures that are present inside it. So, I said this is a vertical section of a mammalian blastula. What are mammals? Mammals are the most advanced group of vertebrates, which includes even us. Okay? And what is a blastula? Blastula is a stage in the development of the embryo of mammals. Okay? So, we all know that all multicellular organisms begin their life as a single cell. This single cell is called the zygote. The zygote is a product of sexual reproduction. When the sperm from the male fuses with the egg from the female, it forms the structure called the zygote, which is a diploid structure. This zygote will undergo various divisions and also will eventually differentiate to become the multicellular organism. In this course of development, this is one stage. Okay, so zygote is the first cell. This cell will first undergo various mitotic divisions called cleavage divisions and become a ball of cells called the morula. Now, this ball of cells called the morula will develop a cavity inside it. Morula is a solid ball of cells where there is no cavity inside. This morula will develop a cavity inside. Once it develops a cavity inside, it is called the blastula. Okay, so this is what we will be learning about in this session. This blastula is covered on the outside. As you can see, it's shown in blue color here on the outside. This is covered by a layer that is known as the zona pellucida. Look at the inside. Here you can see cells are drawn in two different colors. There are pink cells on the inside, and on the outside you can see a layer of yellow cells. Okay, so this blastula, there are two types of cells. The outer layer of cells that are shown here in yellow color are called the trophoblast cells. The cells that are present on the inside, this is the cavity that I was talking about. There are cells present on one side of the cavity that is shown in pink color here. These cells are called the inner cell mass. Now, I will repeat. Outside, the blue colored layer that is surrounding the blastula is called the zona pellucida. The blastula has a cavity inside, which is called the blastocele. You know, coelom is about cavity, so blastocele is a cavity present inside the blastula. And there are two different types of cells shown in two different colors. The outside yellow colored layer is called trophoblast layer. The cells present inside the blastocele is called the inner cell mass. Now, the function of these two cells is also different. The trophoblast layer functions in becoming the extra embryonic membranes as the embryo develops, and this inner cell mass, whatever is shown in pink, is actually going to become the embryo. So, these cells will eventually give rise to what we call the embryo proper. Embryo proper is the organism that is going to finally be formed out of this. Okay, so there's another labeling that you can see here. This is called cells of Robber. What cells of Robber are? See, you can see that it is labeled on the yellow region. So the trophoblast cells here, the trophoblast cells that are covering the inner cell mass. These trophoblast cells that are covering the inner cell mass here, they are called the cells of Robber. The vertical section of the mammalian blastula is a 3-mark question in the practical biology final exams. So what you're required to do here is, you will be given a slide and you will have to identify what is present in the slide. If you're given this slide, you have to identify it as a vertical section of mammalian blastula. If you just write mammalian blastula, you will not be awarded any marks. So for identifying what is present in the slide itself, 1 mark is given. After you have identified what is present in the slide, you are required to write 2 points regarding what you observe in the slide. You can write anything from any point from what I have explained before. Okay, so I will tell you how you can easily identify this slide if it is given to you in the exams. So, you know that I just told you in a blastula, there is a cavity. So, you can make use of that to identify whether it is a blastula. This section will almost be like a circle, and inside, on one side you can see a cavity, and on the other end you can find a mass of cells, which is the inner cell mass. Okay, so if you can find a cavity, empty space within the circle, then you can easily identify it as the VS of mammalian blastula. I repeat, if you identify it as just mammalian blastula, you will not be awarded that 1 mark for identification. It is very important to write what kind of section that is. So you have to mention VS, which means vertical section of mammalian blastula. So let's look at this slide now. Now, let us learn about the vertical section of an ovule. An ovule is also called the megasporangium. This is a part of the female reproductive part of a flower. Ovule is specifically present inside the ovaries. The function of the ovule is to produce megaspores. Since this produces megaspores, it is also known as the megasporangium. The megasporangium is attached to the ovary by a stalk that is known as the funicle or the funiculus. The megasporangium is covered by two structures known as the integuments. Since there are two integuments, we name them as the outer integument. This is the one that is on the outside, and the one on the inside is known as the inner integument. Present inside these integuments, you have a homogeneous tissue that is known as the nucellus. Nucellus consists of many cells, one of which will start looking slightly different from the other nucellar cells. How it looks different is by having a dense cytoplasm and a very prominent nucleus. When you look at the nucellus, you will find all cells similar except for this one cell. This cell would have developed a dense cytoplasm and a prominent nucleus. This cell is called the megaspore mother cell, which will eventually give rise to the megaspores. This megaspore mother cell will undergo meiotic division, which is a reductional division, and as a result, it will form 4 haploid megaspores, which are usually arranged in the form of a linear tetrad. So, these are the two integuments. Within the integument, you have a homogeneous tissue called the nucellus. All of the cells of the nucellus will appear similar except for one. This one cell will have a prominent nucleus and a dense cytoplasm. This cell is called the megaspore mother cell. The megaspore mother cell will undergo meiotic division to give rise to 4 haploid megaspores. Of these 4 haploid megaspores, 3 will degenerate and 1 will remain functional. This one functional haploid megaspore will eventually form the female gametophyte. What do we understand by the word gametophyte? Gametophyte is a structure that will bear the gamete. Okay? So here, if I say that this is going to become the female gametophyte, what I mean is that this is going to form into a structure which will bear the female gamete. I will give you an idea about how it forms. Now I have one cell here, 3 of them have degenerated. This one cell, just the nucleus will undergo division. Okay, here I have shown you one nucleus, here there are 2 nuclei. You know that in cell division, normally the nucleus divides first by a process called karyokinesis, after which the cytoplasm will divide by a process called cytokinesis. The characteristic feature of this kind of division is that initially just the nucleus will divide. After the division of nucleus, there is no cell wall formation. So, what I am trying to say is, karyokinesis is immediately not followed by cytokinesis. So, as a result, you have 2 nuclei lying in the same pool of cytoplasm. Next step, each one of these 2 nuclei will move towards the opposite poles. So, they were here in the center, they will move towards the opposite poles. These divisions are mitotic. Okay, so this is a haploid cell, undergoes mitotic division, only the nucleus divides. Two nuclei are there, they move towards the opposite poles. At the poles, each nucleus will further undergo two more mitotic divisions such that at each pole you have 4 nuclei. After one mitotic division you have 2. Those 2 again undergo mitotic division so that at the poles there are 4 nuclei. Of the 4 nuclei, one nucleus from each pole will migrate to the center again, from here to the center, following which cell walls will be formed. This is called the embryo sac or the female gametophyte. Now, as you can see this in this diagram, this is the outer integument, this is the inner integument. This is the inner integument. So we are not completely fused. The cover is not complete. There is a small pore present here. This pore is known as the micropyle. In the embryo sac, this is exactly what I have shown here. In the embryo sac, the 3 cells that are present towards this micropyle constitute what we call the egg apparatus. The egg apparatus has 3 cells. The central cell here is called the egg cell. On either side of the egg cell, you can find 2 cells that are called the synergids. On the opposite end, this is what is shown here. Here again you can find 3 cells. These 3 haploid cells are called the antipodals. Now there are 2 nuclei present in the center. These 2 migrated from the poles and came to the center, due to which they are called the polar nuclei. So within the nucellus you find the female gametophyte. Female gametophyte has 1, 2, 3, 4, 5, 6, 3 on each. So 6 cells there, and this entire thing is one central cell. So there are 7 cells and 8 nuclei. How are there 8 nuclei? One nucleus in each of these cells present at the poles. In the central cell there are 2 nuclei. Therefore, you can find a 7 cell and 8 nucleate stage here. So this is called the female gametophyte. Alright, so I will repeat it briefly one more time. This is the structure of an ovule. Ovule is attached to the ovary by a stalk called funicle. The body of the ovary is covered by 2 coverings known as integuments. The outer covering is called the outer integument. The inner covering is called the inner integument. The integuments do not fully cover. There is a small space present, small pore-like opening present, that is called the micropyle. Micro means small. So, because it is a small opening, it's called the micropyle. Within the integuments, we have homogeneous mass of tissue known as the nucellus. One of the cells from this nucellus will undergo meiotic division to form 4 haploid megaspores. So, one of these megaspores will eventually become the female gametophyte. Female gametophyte has 7 cells and 8 nuclei, as you can see here. Now we can look at the slide of VS of megasporangium or the ovule. This also is going to be asked, could be asked for 3 marks in your practical exams. Here again you are required to identify the slide, which carries 1 mark, after which you have to write 2 statements regarding whatever you have identified. You could write that the ovule has 2 integuments, there is a nucellus, there is a micropyle and the funicle. You can write those points, any of those points. 2 points will carry 2 marks. Okay, so I will tell you what you can use as a clue to identify this slide when it is given to you in the exams. A micropyle, the pore, I told you that the 2 integuments do not completely cover the ovule, right? So there is a small opening that is called the micropyle. By making use of this, you can identify the micropyle first. Once you see the micropyle like a small slit there, you will be able to identify this slide as the vertical section of megasporangium or the ovule. Again, it's very important for you to remember to mention that it is a vertical section. If you just write megasporangium or the ovule, it will not fetch you any marks. Now let's look at the slide.